So thanks for joining this new start of a new OBON webinar series, which will be introducing some of our newly endorsed projects and giving updates on our existing endorsed projects. And the main idea for these webinars is to have updates provided ahead of our OBON annual meeting, so that at the annual meeting, people attending will have hopefully caught up, either attended in real time or caught up on the recordings of all these presentations ahead of the meeting itself. But it's also a great opportunity to make the broader community aware of what OBON and OBON projects are doing. So it's great to see a lot of new names of people who maybe haven't been particularly involved in OBON so far. So I'm Sophie Siave, I'm the CEO of POGO, and I'm just going to give a brief introduction to OBON, particularly for those who may not already be familiar with it. So I'm just going to try to share my screen. Okay, so POGO is the lead organization for OBON, which is a UN Ocean Decade endorsed program. And this is a link to the OBON strategy, which was published last year. So please visit that URL to find out more about OBON and our strategy. But just to introduce briefly our objectives and vision and mission. The vision is to accelerate informed decision-making to restore the health of our ocean using the universal signatures of life on Earth, biomolecules. And OBON's mission is really to create a global hub for biomolecular measurement of marine life. And we have these four main objectives, which are to build a coastal to open ocean multi-omics biodiversity observing system, which also aims to integrate autonomous sampling and develop new autonomous sampling methods and technologies, to transfer and develop capacity through training programs, but also funded equipment programs for mainly aimed at developing countries. We want to enhance marine omics data systems and ultimately ecosystem models by adding biomolecular components. And we aim to partner with marine conservation and management specialists, policymakers, and the private sector to address sustainable development, which is obviously what the UN Ocean Decade is all about. So this infographic kind of depicts those four objectives and the four pillars of OBON, which are to develop the networks and resources, to innovate through technology development, to enhance information use through, as I mentioned before, models and things like digital twins, and ultimately to inform policy and decision-making for a sustainable future. So we have probably more than 21 projects now, as we're introducing today one of our newest endorsed projects, but we have this big family of projects that are all focusing on biomolecular observations, but several different aspects of these observations. So some of them focus more on the data and the metadata standards and protocols. Some of them are more observatory type projects, time series eDNA observatories. And we have some, particularly from Norway, actually, we have a few projects that are doing really different and novel research using biomolecules, as you will discover in the next presentation. So this map shows the OBON sampling location, so particularly for those projects that are some sort of observatory or that have repeat cruises or one-off cruises that are part of their programs. So this really shows the global scope of OBON, but also some gaps, particularly in the open ocean, where we don't have such good coverage as we do for the coastal zone. So we're really aiming to fill those gaps and to continue to grow the OBON family of projects. In terms of how we're organized, so as I mentioned, POGO is the lead organization, and we provide coordination support through an office hosted by POGO, and Luz is our program officer for OBON, and we also have Fiona, who's the POGO communications officer, supporting OBON activities. In terms of our governance, we have a scientific advisory committee of 22 members from all over the world, and we have a smaller executive committee. And both of these committees are chaired by Margaret Leinen. We're planning to set up a user advisory committee, which will be a really important component of the governance in terms of reaching out to stakeholders and groups that can really benefit from eDNA and biomolecular observations. And in the last year or so, we've set up a number of working groups, which are listed here, and that meet online and also in our in-person annual meetings. We've had a number of workshops focusing on different topics. So we've had a first marinomics technology and innovation, and that should say instrumentation workshop that was hosted by Mbari in California a couple of years ago, and the second one is happening in Hobart next month. We had very recently a workshop on eDNA for fisheries and aquaculture management hosted by FAO in Rome. And I believe it's happening imminently, a workshop on capacity development and eDNA training, but also aiming to set up a regional kind of regional OBON network for Latin America and the Caribbean. And that will be happening at Invermar in Colombia. So that's just a quick overview of OBON and some contact details here and social media for anyone to follow us or get in touch. So thank you very much. We will now move to our invited OBON project presentation, which will be given by Anders Goksøyr. I've been trying to practice the pronunciation from the University of Bergen, who is representing the project with the exciting title of Whales and Polar Bears in a Petri Dish. I think it also has the acronym Marma-detox. So all about toxicology and how biomolecules can tell us about toxicology in marine mammals. So, Anders, if you could please share your screen, that would be great.
Yes, thank you.
Yeah, so we can see the presentation, and it's now in presenter mode, so that's perfect. Thank you. The floor is yours.
Well, thank you very much, Sophie, and thank you to the OBON network for inviting us in, and also, of course, also to the Ocean Decade Committee Office, whatever it's called, for endorsing us into the UN Ocean Decade. Also want to thank the Research Council of Norway, who has funded our project. And if you see this nice photo of a killer whale in the sea, this is taken by one of our partners, Professor Audun Rikardsen at the University of Tromsø, who is also a awarded wildlife photographer. So this project is not only me, but it's a large group of people around the world with partners across the Atlantic. If we start on the left here, we have Jose Vasquez Medina at UC Berkeley. We have Dimitri Spiropoulos at the Medical University of South Carolina in Charleston. And if we go to the red circles, which are the partners, you can start on top where we have Heli Roti, our co-PI from the Norwegian Polar Institute, and her colleagues. We have Pierre Blevin, who is running a partner project called SLICE at Aquaplan Niva, but is also partner in the Marma-detox. And here is Audun Rikardsen at the University of Tromsø. We have Jan Lykke at the Norwegian Life Science University at Ås. And here in Bergen, we are my colleagues André Ficadu, postdocs Sophia and Bram, and PhD student Sarah, and Maud, who's actually situated in Leuven in Belgium, with Kathy Debier, who is one of our scientific advisory committee members, together with Bjørn Winre Jensen from Trondheim and Magali Hood from Canada. And we have partners in Portugal, Felipe Castro and Raquel Rivo at CIMAR, University of Porto, and Cinta Porte at the CCG Institute IDEA. So the background is, of course, that marine mammals are exposed to bioaccumulating and biomagnifying contaminants. And they are actually threatened by these compounds because they biomagnify to very significant amounts especially in the top predators. And here we are talking about many different types of compounds. We have a group called persistent organic pollutants, POPs, but we also have something called contaminants of emerging concerns, or CECs, where you can find the plasticizers like phthalates and bisphenols and other endocrine disruptors. So we know that these compounds are toxic and could disturb the organism's physiology, immune system, reproduction, etc. But how this works is something we would like to know and find out more in detail. The results of this biomagnification is this. Here you can see that sperm whale, pilot whale, killer whale are the top most polluted animals probably on the planet. The beluga or white whale and the polar bear are next. This is a log scale, as you notice. And then some of the more mid-trophic racers, like the minke whale, the fin whale, humpback, and blue whale at the bottom, have much lower levels of these pollutants. For comparison, humans are possibly at this level, depending on their lifestyle and what they eat. We know that Inuits in Greenland also are eating stuff with high levels of contaminants and end up in this range. So what is the effect of this, and what is the cause of these sometimes large differences, like when we compare killer whale and polar bear? They eat more or less at the same level of the trophic chain. They should be having the same levels of contaminants, and for some contaminants they are very similar, but for other contaminants, polar bear is quite much lower, which is an interesting observation. So what happens when these contaminants end up in the bodies is that they are biotransformed, as we call it. It's a metabolization process. These compounds enter mostly through the food ingested, and they are distributed to the liver, where what we call detoxification and biotransformation takes place. In two phases where enzymes involved convert the very lipophilic contaminants to more water-soluble metabolites that can be eliminated from the body. This is actually a three-phase process because the enzymes in the phase 1, they add a reactive molecule to the contaminants, normally an oxygen, and this is conjugated to an endogenous molecule, which can be sugars or amino acids or sulfates. And this is obviously a more water-soluble molecule, but to get this molecule out of the cell, we need a phase 3, a transporter activity. So we have transporter proteins that are able to push these molecules out of the cell and then for excretion. And this process is, of course, different in different animals. We don't know too much about that in marine mammals. And if you want to study this with other animals, we normally do some in vivo experimentation. With marine mammals, this is, of course, not feasible and also ethically not acceptable. You can do something with the beaching animals like the elephant seal here, where you can approach it and get some blood samples or small biopsies. But for the other animals, this is a tricky thing to do. And of course, you can do some in silico population modeling, ecosystem modeling, etc. What you can do if you have more specific samples, like from stranded animals or with other animals, you can do in vitro activities, measurements of enzyme activities, levels, transcripts, and you can model activities and also do modeling of protein ligand interactions. I started my work on marine mammals with the samples of minke whale liver when I was out as a whaling inspector. I was able to get liver samples and had liquid nitrogen. I could bring my samples back to the lab and I could do this as part of my master thesis. So what we are doing now is to make this in a more middle out approach, establishing cell cultures or using biopsies and tissue cultures with the aim at some point to develop organoids which more resemble the organs in the body that are responsible for the toxicological response. And with these systems, we can do exposures, we can do multi-omics data analysis, we can do dynamic modeling, and we can do genome mining of these animals. And with the genomes and the dynamic processes mapped, we can also do genome scale metabolic models at some point. And of course, we can research how the effects of stressors impact the processes we are looking at. This approach here was presented in a paper last year that we developed during a workshop in Berkeley. I had a Fulbright scholarship staying with Jose Vasquez Medina, and we invited many of the researchers in the field to discuss how this should be approached. And we got this published last year in Frontiers in Marine Science. So I wanted to show you a quite short video. It's about 6 minutes, and I hope you will be able to hear the sound.
Large marine mammals are some of the most polluted animals on the planet. This is a result of a process known as biomagnification, where chemical pollutants get ingested by small marine organisms and build up in the fatty tissues of larger animals as they move up the food chain. Animals at the top of the chain, such as killer whales, can end up carrying over 100 times the harmful level of pollutants, enough to classify them as toxic waste. Chemical pollutants can have deadly effects on the health and survival of these animals, making them a major threat to these species.
Well, we know that these chemicals can impair the animal's ability to reproduce and to handle infections and disease. What we know much less about are the specific internal mechanisms that cause these things to happen.
Studying the effects of pollution on marine mammals is a challenge. These animals are widely distributed across the world's oceans, so finding them can be difficult. In addition, scientists need samples from organs like the liver, kidneys, and gonads. These are places where pollutants tend to build up, but getting organ samples from wild animals is nearly impossible. How do you study the effects of pollution on large marine mammals in a way that's both humane and effective?
The Marma-detox project is an international research project that aims to address all of these challenges using cell cultures. Our goal is to understand the effects of ocean pollutants on large marine mammals in a way that is efficient for the researcher and ethical for the animal. To do this, we partner with another research project called SLICE, who takes small skin and blubber biopsies from whales in the wild. We then use skin pieces from those biopsies to create cell cultures. We can use these cells to study effects of pollution in marine mammals in ways we never could before.
Marma-detox and SLICE are sister projects in that we are both trying to understand the specific effect of chemical pollution on large marine mammals. In the SLICE project, we want to focus on the tissue level by keeping alive blubber slices to further expose them to pollutants and stress hormones. To obtain our sample, we use DART to take small biopsy from wild whales, just below their dorsal fin. So far, we have been able to collect biopsy from fin whale, humpback whale, and killer whale. After we collect the biopsy, we store them in a sterile container and ship them to the lab for experimentation.
Our combined labs are currently conducting 3 different types of experiments. So the first is the isolation and establishment of the whale cell cultures, which is done by the Marma-detox. So we grow the cell cultures and then expose them to different levels of mixtures of pollutants to study how these mixtures affect the cell's DNA and cellular processes.
So here we are growing killer whale cells, fibroblasts, from skin biopsies, and they're looking good.
SLICE conducts experiments to test how pollutants affect large marine mammals at the tissue level. So they do this by looking at how pollutants affect groups of different cells, rather than just one cell type, like in the Marma-detox.
We all know that pollution has very bad impacts on the environment and mammals in general, but actually we don't really know the specific effects of pollution. So we would like to know what's going on and in which way we will be affected by pollution. So our project will focus on doing transcriptomic analysis and to try to better understand which genes will be increased in terms of transcription or decrease due to the exposure of pollutants. Thanks to this insight, we might be able to take actions to try to better protect those threatened mammal species and hope for a better future.
So once the biopsies arrive at the lab, they immediately cut these into very thin slices of blubber. So in the SLICE project, the slices are exposed to the mixture of pollutants and cortisol, which is a type of stress hormone. And they do this to study how the slices respond to a combination of both pollution and stress. So Marma-detox and SLICE are together optimizing the usage of each biopsy, where SLICE uses the blubber for slicing, and Marma-detox uses the skin part for establishing cell cultures. So the nice thing about our research method is that we can obtain our samples without bothering the whales, well, too much. While the downside of this method is that the range of cell types we can obtain is limited to the cells found in skin and blubber tissue. So these cells don't express all the response pathways we want to study. Ideally, we want to study how pollutants are affecting organs like liver, brain, and fat tissue, for example. But these are tissues we can only obtain from dead whales. So instead, we are trying to reprogram the cells we have into other cell types, such as liver cells and fat cells. We might even be able to reprogram them into brain cells with some help of collaborators in Italy. I mean, if we're successful with this process, the research possibilities will be endless.
The biggest advantage about working with cell cultures is that these cells can be maintained and multiplied to large numbers in the right conditions. The methods we try to develop will make our research much more convenient and affordable than if we were to study these animals in the wild. this kind of research is difficult to perform, and you need the combined resources of several partners. So we are very excited to be paving the way for toxicology research on marine mammals.
Marma-detox and SLICE are projects that could potentially play a key role in helping to create cleaner oceans. The findings from their research will help us understand and beat marine pollution, a major challenge that the UN is looking to address by 2030 through their Decade of Ocean Science for Sustainable Development. By providing important knowledge about how pollution affects marine mammals, Marma-detox and SLICE can help create solutions and policies that would reduce or ban chemical pollutants from entering the ocean. Through their innovative research, the Marma-detox and SLICE projects hope to build a more sustainable future for our planet by supporting healthier marine mammal populations and a cleaner ocean for us all.
Yes. Thank you. Well. So back to the project. So the main objectives of our project, they are to establish fundamental knowledge about the genomic basis of these defense systems in selected marine mammal species, to characterize the functional properties of key molecular targets for environmental contaminants, and to characterize responses in marine mammal cell cultures, stem cells, and fibroblast cell lines, and also to integrate in silico and in vitro results with available reports or data on associations between contaminant exposures, biomarkers, and omics data from free-ranging animals. So far, after 3, I guess, busy field seasons with the SLICE project in command, we have obtained a lot of samples, and Sophia here on the left side, she has been working hard whenever there is a sample coming in, as you saw in the video, to culture cells, both the fibroblasts and the adipocyte-derived stem cells, ASCs. And we've now had samples from both polar bear, sperm whale, killer whale, pilot whale, humpback whale, and the fin whale. The polar bear was taken from a cruise with the research vessel Krontmenshaken in the spring, which was a very successful cruise, and I would have liked to show a video of that specifically, but there's no time for that now. But at least we have now a number of fibroblast cultures in our bank, also ASCs, and also a number of biopsies that can still be used for different purposes. And when the fibroblasts are cultured and grown in the lab, we can use those for exposure experiments. And so far we have done experiments with both the killer whale and the fin whale and the polar bear for POP, that's pollutant mixtures. And we've sampled RNA to do qPCR, starting to do RNA sequencing, and also to look for other omics markers in these exposure situations. But as Sophia mentioned in the video, we are also wanting to obtain other cell types. And so she said liver cells or nerve cells. And there is the potential, if you have ASCs or MSC, mesenchymal stem cells, to be able to differentiate these into other cell types. So that's one thing we have been working with in the lab, both here and with partners, as Sophia mentioned. And we also have Dimitri in Charleston. And I will show you a few data on this. Maud and Sophia in Bergen has been looking at the transformation to adipocytes through adipogenesis. And Dimitri in Charleston has been working hard on different approaches, one of them towards nerve cell differentiation. So first, Maud and Sophia tested different cocktails and protocols for initiating adipogenesis, so changing fibroblasts or ASCs into adipocytes, pre-adipocytes or adipocytes. Mature adipocytes can be stained with the old red O or adipored stain, which turns them into red cells which stains the lipid in the cells. And of course, these cells, as you see, look to be very successfully transformed into adipocytes. At least they are adipocyte-like, which means that with polar bear cells, we are able to do this. The protocols work, and we can initiate adipogenesis. However, these protocols did not work with any of the whale cells that we have tested so far. So that's one issue to learn from. There is something different in the way the whales handle this differentiation cocktail and how they control their own differentiation of adipocytes in their blubber tissue. And then the other approach that the petri has been testing out. Well, he has tested out quite a lot of many different approaches. What seems to be giving some interesting results now is to differentiate fibroblasts into neuronal-like cells. And this seems to be in a neuroepithelium type of cells, which is very promising for doing more research on both neurogenesis, but also on how toxicants can affect the neurosystem, which is an important aspect of toxicology. Now I would like to turn to the more genome side of the thing. And we have something called the chemical defensome. The defensome is the part of the genome that comprises the genes involved in the defense of the organism, which includes transcription factors, regulating the expression of genes in the biotransformation pathways, and the transporters, and also stress response genes. We recently introduced a term we call the extended chemical defensome, which also includes avoidance behavior. But so far, we will treat that as a black box. Within this set of genes in the genome, we have around 400 to 500 genes in most of the organisms that have been studied so far. And characterizing this defensome can give us insights into the potential, both the potential to handle contaminants, but also the potential susceptibility of organisms to pollution. And with Bram here as a postdoc in this project, also linked to the Earth Biogenome project in Norway, he set up a pipeline to find the genes in the different marine mammals and in their relatives. I'm not going into the specific parts of the pipeline, but we can see here on the spider plot to the right how the orange-colored numbers here represent the artiodactyls, so the land-living terrestrial relatives of marine of cetaceans. So whales in blue, the baleen whales in light blue, and toothed whales in the darker blue. We can see that there is much more of the genes in the oxygenases, in the reductases, and in the transporters. So there is a loss of genes in the marine mammals. And within the oxygenases and reductases, we find specific phase 1 and phase 2 gene families. And again, a lot more genes in the artiodactyls, terrestrial mammals, than in the marine mammals. And if we go more specifically into subfamilies of genes in the cytochrome P450 gene family or superfamily, we can see here, these are the cetaceans. These are the relatives, the artiodactyls. And we can see that the darker the color, the less genes are present in these animals here. There is several examples where these subfamilies of genes have been eroded away in the marine mammals. Also over here. So both the CYP4, CYP3, and CYP2, CYP3, and CYP4 subfamilies, many of those have been eroded and have lost genes. We also know the same is true for transferase genes in the phase 2 activities. And we know that cetaceans have lost some of the transcription factors, specifically the PXR and the CAR transcription factors, for those who are more familiar with these terms. And even a more detailed genome investigation we performed. It actually started before the Marma-detox project, but it was finished 2 years ago, into the genome of the blue whale. Starting with sampling outside Svalbard, north of northern Norway. Heli and the Polar Institute people were out sampling biopsies. We got in touch with the people in Canada at the Royal Ontario Museum, who were working also on the blue whale samples from a stranded blue whale in an iceberg. And we were able to collect a number of samples, both from Norway and from Canada, in addition to historical samples from museum skeletons. So here from sampling from a blue whale skeleton from 1876 at the University Museum of Bergen, drilling in to get bone samples that were sent to Canada for sequencing. And the story ends with some interesting new knowledge about the blue whale and the genome, which was published 2 years ago with our Canadian collaborators. Just a month or so after another blue whale genome was published. This was based on a Pacific blue whale, whereas ours was a North Atlantic study. And the findings indicated actually that there had been some interbreeding between the blue and the fin whale. So that when we did a d-statistics of the introgression and the gene flow, indicated there had been both a very recent hybrid, so this one, indicating that this is an offspring of such a mating, whereas the other are more offspring through several generations, indicating that these offspring are actually, so the first generation offspring are actually fertile, reproductively fertile. So actually all present-day North Atlantic blue whales showed significant female introgression. But also 4 of the 6 historical samples showed introgression. None of the hybrids carried female mitochondria, so obviously their mothers were blue. And this showed a unidirectional gene flow from the fin into the blue whale gene. All in all, fin whale sequences constituted about 3.5% of the genomes of these North Atlantic blue whales, which is about the same as Neanderthal genes are present in our human gene. Just to summarize how this defensome part of the genome can help us in across different fields of environmental toxicology, including mixture toxicity, species sensitivity, and maybe even explain biodiversity in some cases, is because these gene sets are important in handling toxicity and exposure in the environment. Finally, I'd like to just say a few words about our outreach activities. We had Audrey visiting. I think she's in the audience actually. Audrey was a Fulbright student visiting us from the US, and she took very well care of preparing both the exhibit about Elida. She is a killer whale, or was a killer whale baby, 3 years old, which had significantly high levels of pollutants, but was found dead, apparently caused by this pollution load. And using her examples in an exhibit, we tried to put focus on ocean pollution and how these pollutants affect animals like the marine mammals that are biomagnifying these compounds. It was both a well-visited exhibit at the museum, but also on a research day at the center of Bergen, and we got the first page in the newspaper. And we still have Elida in her coffin. The Elida model and the coffin was made by a student from the arts department at the Faculty of Arts at the university. It's still sitting here in the lobby entrance of our department. And I was also positively surprised that finally this summer, after many years of waiting, the UN established a science policy panel on chemicals, waste, and pollution. So in addition to the IPCC and IPBES that we already had, now the third global crisis also has its panel from the UN. And with that, I'd like to thank you all for listening, and thanks to the people who have been involved and who have been supporting us through these years. We are actually halfway through our project, starting up as a new OBON project. I think we applied almost a year ago. It took a while, but the project started 2 years ago soon, and so we are almost halfway in our project. Thank you for listening.
Okay, thank you very much, Anders. I've hadn't heard about the this new intergovernmental panel. Is it new?
Nobody heard about it. It has been so little announced.
Yeah.
It came out in June.
Okay.
See, they haven't had their first kind of official meeting, but I mean, this website was from this summer, and even in Norway, we didn't know much about it.
Okay, yeah, I'll definitely look into that. So I guess they'll also be producing assessments every few years, or
yeah, let's hope they will work in the same way as the other ones.
Yeah. Okay. Are there any questions? I have another one, but I want to give opportunity to anyone else.
Stop sharing, maybe.
Yeah, it's a shame that we couldn't have the presentation from Courtney. I don't know if you know Courtney War from what's her affiliation?
Yeah, the University of Bodø, Nord University.
Yes, Nord.
We have actually collaborated with her.
Okay, that was going to be my question. It seemed like there's potential for a lot of complementarity between your two approaches to non-invasive marine mammal investigations.
So I could show a slide that our Sophia prepared from her student Helena, using fibroblast cells from killer whale that Sophia here had prepared to test how viral infection affected the response, or actually the other way around, how pollutants affected the response to viral infections.
Okay, so yeah, so sounds like you're working at the cutting edge of developing these new methodologies.
Yes, and we have had a lot of approaches to have samples like cells and samples sent, and of course we are happy to share, but of course we also need to prioritize PhD students and postdocs who need to get their stuff out before we send everything away. But we are very interested in contributing to expanding this field of research.
Yeah, well, that's great. Okay, I don't see any other questions. So in that case, I would like to thank you again, Anders, for taking the time for this presentation, and it will be, the recording will be uploaded to the OBON YouTube channel, and we'll make sure that all our other projects watch the video before our annual meeting. So it was definitely very useful to have this introduction. So yeah, thanks again, and we look forward to interacting with you. The annual meeting will be, obviously it's in Hobart, so the time zone won't be great for people based in Europe, but there will be a remote participation opportunity. So if there are, times at either end of the day that that would work for you, I'll make sure that we share the links.
Yeah, oh, that's great.
Okay, well, thanks very much to those who joined, and thank you again, Anders, and we look forward to continuing to the collaboration through OBON.
Thank you.